Hello, everyone, and welcome to today's webinar hosted by Jen and sponsored by Oxford Nanopore Technologies. Today's topic is uncovering RNA splicing cell-specific impact on clonal blood disorders with GoT-Splice. Thanks so much for being here. I'm Urak Thomas, Senior Editor with Jen, and it's my pleasure to be your host for today's webinar. Our speaker today is Dr. Mariela Cortés López, a postdoctoral associate at Weill Cornell Medicine and New York Genome Center. Before we get started with the presentation, let's go over some background on today's topic. The utility of long reads is well documented in studying complex genomic regions, structural variation, epigenetic modifications, and transcriptomics. Specifically, long-read data are increasing our understanding of RNA splicing factors. Although the regulation of splicing factors affects hematopoiesis, the role of dysregulated RNA splicing in clonal blood disorders, such as myelodysplastic syndromes and other blood cancers, remains unclear. In this webinar, Dr. Cortés López will present a novel approach, GoT-Splice, developed by the Landau Lab at the New York Genome Center in collaboration with the Abdil Wahab Lab at Memorial Sloan Kettering. This method integrates genotyping of transcriptomes with Oxford Nanopore long-read single-cell transcriptome profiling and proteogenomics. GoT-Splice comprehensively profiles transcriptomes, surface proteins, somatic mutations, and full-length RNA splicing at single-cell resolution. During the webinar, you'll hear a case study where GoT-Splice was used to study hematopoietic progenitors from MDS patients with mutations in the splicing factor SF3B1. And you'll also hear about the findings that collectively demonstrate the power of GoT-Splice in delineating the cell type specific impact of somatic mutations on RNA splicing. Today's presentation will be followed by a live Q&A session. If you have a question for our speaker, you can enter it at any time in the Ask a Question panel on your screen. All right, let's get started.
Thanks for the introduction, and hello everyone. As mentioned before, I'm going to talk to you today about a story that we recently published where we try to look into the specific impact of clonal blood disorders, but with a new technique that we recently developed in the Landau Lab. So to start, let me remind you that perhaps you're not aware, but we are mosaics. So when we are born, our genomes look the same, but with the accumulation of age, we also accumulate mutations. And these mutations can appear in different cells at different rates, different times. And some of them are just passenger mutations, but others could then, with the passing of the years, becoming more frequent, create these clonal expansions, and then result in the development of hematopoietic diseases, for instance, in the blood. Some of those mutations are frequent in certain genes, and recent studies have allowed us to understand the frequency of those mutations and the genes that they tend to target. And one of the most interesting genes, gene classes that I've been looking into my studies has been splicing factors. Splicing factors have been reported to acquire a variety of mutations, and many of them tend to appear in certain hotspots. Some of them also have a gain of function. They are changing the protein in a way that they completely change their function. And this is important because alternative splicing has also been reported to be involved in many of those hallmarks of cancer that we observe. So many alternative splicing events result into important processes that allow cancerous cells to progress. Especially in blood cancers, what we see is that there is an increased frequency of splicing factor mutations. In some of the variations of certain blood diseases, we even see an accumulation of more than 60% of the patients presenting a particular mutation. In this case, one of the most interesting ones is perhaps the core splicing factor SF3B1, but there is also other splicing factors which are part of these core proteins that are required to do the splicing function, which are also highly mutated. So in particularly, SF3B1 tends to have hotspot mutations in some of the the domains. More significantly, this K700 mutation is one of the mutations that we have been focusing on our study. SF3B1, the consequence that this mutation has for the protein is that will increase the usage of alternative 3' splice sites. What I mean for that is that normally during the process of splicing, we remove this middle intronic sequence. However, when we have an aberrant splicing mutation in the SF3B1 gene, what results is that we have a protein that then recognizes an aberrant branch point. And this aberrant recognition of the branch point will result then into the inclusion of part of this intron that should be removed. And when this happens is that we get these different transcripts that will have a fragment of the intron, and some of them will lead to the progression of nonsense mediated decay, or will generate aberrant proteins. So we wanted to study the relationship between splicing aberrations and the cellular phenotypes that we have observed. However, there have been many pioneer studies that have tried to look into this, but they have been limited by multiple factors. First, in the cells that we can get in the samples, like blood samples that we can get of patients, there is a high cell heterogeneity. And this is usually missed when we do bulk sequencing. And this has also led to many researchers trying to look into at the single cell level. However, at the single cell level, we also have complex cellular phenotypes, which sometimes are difficult to characterize. And moreover, at the single cell level, we still rely on short-read reconstructions, which sometimes when we want to look at the transcriptome level, do not allow us to specifically identify the isoforms that might be being affected by this specific change in the mutant SF3B1 phenotypes. So in the lab, we have been taking an approach to look at different aspects of cancer evolution in using a multi-omics perspective. What we mean by that is that we look at different levels in single cells. So we can, in this case, focusing on the cells that might have this SF3B1 mutation, but also their transcriptomes. But how do we characterize these mutations? So previously in the lab, some of my colleagues developed this technique called genotype of the transcriptomes, or GoT for short. GoT relies on the commercial 10x genomics assay, which then allows for the generation of single cell reads. The single cell reads are then amplified using a specific primer targeted to the mutation that we are interested on. And so then we can capture this mutation in higher numbers. And because we have these cell barcodes will allow us to then go back and see where this particular mutation was, if it was present in a specific cell or not. And this is combined with the original library, and everything can be integrated. So this was a very cool advance. However, it was still relying on the short-read sequencing. But recent advances also in long-read sequencing will allow us to expand this GOT framework. And in a recent published study, GoT-Splice, we describe how we can combine GOT with other technologies to make it more precise and help us to understand this characterization of complex samples. So in this case, in GoT-Splice, what we have is we start with the same 10x genomics platform where we get the single cell information. And from this single cell library, instead of going directly to do the fragmentation that proceeds before getting the Illumina sequencing, what we do is that we use Oxford Nanopore sequencing in order to get the long-read transcriptome. And part of the library is also, it goes through the process of GOT. So we amplify a particular loss type of interest, in this case, SF3B1. And at the same time, we combine this with proteogenomics characterization, which will allow us to then distinguish specific cell types. So for this, we use the previously developed in the Genome Center CITE-seq technology. And at the end of this library construction, what we will get is from a single cell, the alternative splicing, the transcriptome and the protein markers, but also the genotyping. So we started this study with MDS cohort. So MDS is myelodysplastic syndrome. And this MDS cohort was composed by 3 individuals with an SF3B1 mutation. So let me now tell you a little bit of what we found. So from this study, these 3 initial patients, what we saw is that we were able to recover the expected cell types that we were looking at in this blood samples. So what we got from this study is actually the expected cell types that we wanted to, that we often observe in hematopoietic progenitors. Interestingly, another observation of our study was that we see actually that now with this information of the genotyping, that most of the mutant cells tend to appear in the megakaryocyte erythroid lineage, meaning that they seem to accumulate in the erythroid progenitors. And this can be really quantified in the sample where we see a higher density of the mutant cells and their erythroid progenitors. With having the cell surface characterization of the sample, we also were able to assess the specific identity of the cells. And so we have the cells that were validated both by RNA, but also by the cell surface markers. And interestingly, combining this with the genotyping gave us a view of certain markers that seem to be slightly more specific for the mutant cells. And moreover, the presence of these markers also seems to correlate with the differentiation trajectory, with some of the particular markers for the erythroid progenitors appearing later on the trajectory. Moreover, these erythroid progenitor cells tend to accumulate these changes in the cell cycle and also translation. So when we just did a comparison of the mutant cells, mutant erythroid progenitor cells versus the wild erythroid progenitor cells, what we observe is that in most of the mutant cells, we have an increase of genes that are associated to translation functions and also cell cycle. And these genes, some of them are even involved in the TP53 pathway. And as we can observe here, they also appear differentially fractions with the passing of the different progenitor trajectory. now having this information, all that I showed you before has demonstrated the applicability of having the genotyping information and also the cell surface information. But what happens when we want to go to the splicing phenotype, where we especially were trying to capture these full-length transcripts? So interestingly, in order to achieve this goal, we realized that actually adding a biotin pull down step in the preparation of a library increased the capture of proper RNA transcripts, sometimes even achieving yields above 80% of proper transcripts capture. And having this long-read sequencing information at the single cell level then gives us different advantages. Because normally, just having short-read sequencing, we will have small fragment that only allow us to see the end of the transcript. However, and when we have the long-read sequencing, then we can see which are the isoforms that could be changing across multiple cells, but also which are the isoforms that within the same cell identified by the cell barcodes are behaving differently. And this is just an example of the degree of increased information that we gain now with the long-read sequencing. So in this case, for this gene, we can observe that the coverage of that particular cryptic splicing event, which is a characteristic of the SF3B1 mutations, is only highly captured with the long-read sequencing. Moreover, we can now go and look into the different alternative splicing events because we have a better capture of many of those genes. And we were able to recapitulate the phenotype that is that has been described in SF3B1 mutant. So here we see that SF3B1 mutant cells tend to have a lot of cryptic 3' splice sites. And this is just an example of one of those cryptic splice sites, which appears only in the mutant cells. And now having this resolution of particular genes that have this cryptic splicing, we can look into the continuum of erythropoiesis and see how with the increase of differentiation, we also observe different patterns of the splicing of particular genes, some of those genes being more involved with processes that are critical for hematopoietic cells and erythroprogenitors, such as the heme metabolism, or also the development of the erythroprogenitors. And more interestingly, we can also observe certain isoforms that are only specific for certain cell types. And in this case, I just wanted to highlight some this cluster of genes that are also only present in the erythroprogenitors. And as we can see from the labels, most of those genes tend to be involved in erythroid differentiation, making sense that certain certain genes that are important for erythroid differentiation are also specifically misspliced in erythroprogenitors. Moreover, we took another group, independent group, as our validation study for the genes that we were capturing here. And in this case, we were able to show that this is not only, the events that we were capturing were not only a handful of events that happen in a single cohort, but also they tend to be conserved across the other patients with the same type of mutation. And some of the events have interesting functions. I will come back to this in a couple of slides. So exploring the phenotypes, so far we've been looking at the disease state, But this technology can also be applied before the disease appears. Especially going back to the abundance of these clones which accumulate with age, there has been a lot of studies where it has been reported of how many mutations tend to appear constantly with the increase of age. And some of them just in normal individuals before they develop a particular disease tend also to be splicing factors. So here we see that in normal individuals with this clonal outgrowths, they already present a high frequency of SF3B1 mutations, but also SRSF2 or U2AF1, which are other particularly interesting splicing factors. And then they sometimes also represent a higher risk for patients to acquire, to develop a clonal disease after some years. So GoT-Splice, we decided to apply it to a clonal hematopoiesis group. In this case, this group was a little bit smaller, only 2 individuals with different mutations in SF3B1. But in these individuals, we were also able to recover all the different hematopoietic progenitor populations, similar to the ones that we detected in MDS. Here also, striking observation that we got was that erythroprogenitors tend to also be the ones that accumulate more mutants. This was also measured through the pseudo time. So when we get an increase in the trajectory of differentiation, we also see an increase of the mutant cell ratio. Moreover, differential splicing genes affected in SF3B1 mutant cells, they also tend to be shared with the ones that we observed in MDS. And they also present this phenotype of being alternative splicing events which lead to cryptic 3' splice site change, which is characteristic of SF3B1. So here I'm highlighting some of the cryptic junctions that we were able to recapitulate in this pre-malignant state and also MDS. And perhaps one of the most interesting examples of this that we highlighted in the paper was the missplicing of the apoptotic genes. In this case, a particular apoptotic gene is BAX. And we observed that BAX has actually several isoforms, but particularly BAX omega seems to be a protective isoform. So when BAX omega is generated, cells tend to avoid going to apoptosis. And we see an increase on the generation of BAX omega in relationship with the trajectory. Going further up to the trajectory, then we see more erythroprogenitors and we see a higher level of alternative splicing for BAX, generating more BAX omega. And moreover, with the help of some of our collaborators, we were able to indeed assess that BAX omega has an anti-apoptotic role, which could explain why we see an accumulation of erythroprogenitor cells, but they don't go into a massive apoptosis. And not only GoT-Splice can be limited to only to study splicing factors like SF3B1, but we can also look at different splicing factors and even other regulators. So one final takeaway from the paper is that we were also able to recover to genotype and also to recover the splicing patterns of U2AF1 mutations in this way, in this particular example, we were looking at an AML sample, and we were able to genotype a fair amount of wild type versus mutant cells. And those mutant cells tend to present mostly exon skipping events, which are characteristic of the U2AF1 mutant phenotype. And on the other hand, when looking at a different gene, which is not associated with directly with the process of splicing, but rather an epigenetic regulator, DNMT3A, what we observe is that we were also able to recapitulate specific splicing changes, in this case, SRSF3, which is also a splicing factor, meaning that GoT-Splice could also be a technique that would allow us to understand how other type of regulators or the type of genes can also be involved in the different processes to regulate alternative splicing within single cells. So in conclusion, GoT-Splice is a technique that allows for the profiling of the single cell level of genotype and expression, surface markers, but also splicing. And SF3B1 mutations tend to be very specific to certain cell types, in this case, some erythroid progenitors. But moreover, the impact of these SF3B1 mutations can be manifest before the disease onset. And this is important because this will mean that there are certain genes that are key for this conserved mutational impact that we, the phenotypes that are being present already before the disease arises. And with that, I would like to conclude my talk, and I invite you to look into our study. Our protocol is coming up very soon this year, and also I'm happy to take any questions.